So I'm going to talk about sleep. Sleep is an essential innate behavior. We know how to sleep even before we're born. And as far as we can tell, all animals sleep, including mammals, birds, fish, flies, and worms, and even jellyfish who don't even have a centralized nervous system. And in fact, jellyfish sleep was discovered at Caltech just a few years ago. So for non-mammalian animals, sleep is primarily detected by a lack of movement. So to score a sleep episode, the animal has to stay still for a certain amount of time. And if you apply mild sensory stimulation, they should show no motor response. So for mammalian sleep studies, we generally measure EEG and EMG. So EMG measures skeletal muscle tone and reflects somatic motor activity. The EEG measures brain state, and as Emery talked about, in general, the so-called desynchronized EEG, low amplitude, high frequency, it's associated with heightened conscious experience, which happens during both wakefulness and rapid eye movement sleep when we experience vivid dreams. And in contrast, the slow wave activity that you see during non-REM sleep is associated with a lack of conscious experience. So for humans, we measure EEG and EMG and also a few other things like the heart rate and breathing and so forth. And these sort of autonomic motor activity also slows down. So the point is that falling asleep is not just about changing the state of consciousness that you can measure with EEG. It's also about a reduction of both the somatic activity, motor activity that you measure with EMG, and also the autonomic motor activity. So my lab is interested in the neural circuits controlling sleep. And what I hope to convince you today is that a lot of the neurons that promote sleep are actually part of the neural circuits controlling these two types of motor activity, and hence the title of my talk. So as Emery mentioned, the arousal system, a lot of it resides in the brainstem and posterior hypothalamus. So the well-known ones include some monoaminergic neurons like dopamine that Emery talked about, noradrenaline, serotonin, histamine, and cholinergic neurons in both the brainstem and the basal forebrain, and also some peptidergic neurons, the most famous one being the orexin neurons in the hypothalamus. In contrast, we know less about the sleep circuit. According to the textbook, there is a single sleep center in the brain, in the mammalian brain, and it's called the preoptic area in the anterior hypothalamus. So the idea is that the preoptic area, the POA, contains some GABAergic neurons that promote sleep by inhibiting a lot of these well-known wake-promoting neurons. And indeed, some years ago, by using retrograde labeling and gene profiling, we were able to identify a subset of the POA neurons that promote sleep. But it turns out that this is not the whole story. Because about 4 or 5 years ago, we and others have also seen some sleep promoting neurons outside of the POA. So that suggests that the sleep control network is actually much more distributed than this textbook model. So we decided to perform a whole brain screening for sleep neurons. We have two basic criteria for sleep neurons. First, they need to be sleep promoting. Their activation needs to increase sleep and their inactivation should decrease sleep. And second, they should be sleep active so that they need to be active at the right time to do their job. And for screening, we also have two corresponding strategies. We could either screen for sleep promoting neurons and then see if they're also sleep active, or we can screen for sleep active neurons and then see if they're also sleep promoting. So today I'd like to take you through an example of each strategy. So to screen for sleep active neurons, we use this technique developed by Li Chunluo's lab. It's called FOS trap. So we cross two mouse lines together. One of them expresses CRE-ER under the FOS promoter. So we know that FOS is an activity-dependent early gene, immediate early gene, so that the active neurons should turn on FOS and therefore express CRE-ER in this mouse. And the other reporter mouse expresses fluorescent protein, in this case GFP, in a CRE-dependent manner. So we divided these cross mice into two groups. One of them we call the SD, sleep-deprived mice. What we did was to pair the injection of tamoxifen with 6 hours of sleep deprivation. So the idea is that the wake active neurons that are active during the wake period, right, sleep deprivation, should express CRE-ER, and tamoxifen should cause the CRE-ER to enter the nucleus and cause permanent labeling of the wake active neurons. And then the other group, we call the RS, recovery sleep group. So we first sleep deprive them and then inject tamoxifen when they're in this recovery sleep period to label the sleep neurons. So this was done as a collaboration between Zhe Zhang and Peng Zhong, former postdocs in my lab. So when we compared these two groups of mice, we saw multiple regions with different expression of GAP between the two groups. Now, if you focus on this bar here with the highest relative expression of GAP in the recovery sleep group, it's shown here. So it's basically the ventral region of the periaqueductal gray, the PAG. So the PAG is this sort of inverted pear-shaped structure, and this is the aqueduct. So you can see that there are a lot more GAP neurons in the RS group than the SD group. So next we wanted to know who these GAP neurons are, because clearly they're just a subset of the neurons in this region. And for that, we did gene profiling. So the reporter mouse that we used expresses not only GAP, but also this ribosomal protein called L10A. So if we use an antibody to pull down against GAP to pull down L10A, we can also pull down the mRNA attached to L10A, because this ribosomal protein is normally attached to mRNA for protein synthesis. So we can sequence the attached mRNA. And so this is what they call the volcano plot. Each dot is a gene. This axis is the relative expression of that gene in the putative sleep active neurons labeled by GAP relative to this entire chunk of tissue. And this is the p-value. So if you zoom into this red box here with the highly significantly enriched genes, there's obviously a bunch of them, but for both scientific and practical reasons, we decided to focus on neuropeptides. So among the 3 neuropeptides in this box, we found that this gene CALCA, which is calcitonin gene-related peptide alpha, it shows a high overlap with the GAP-labeled putative sleep neurons. So we decided to focus on the CALCA population. First, we wanted to make sure that these neurons are really sleep active, because even though the trap technique is supposed to label the sleep active neurons, CFOS is not a perfect indicator of activity. It's also affected by other things. So for that, we did directed recording. What we did was to tag the CALCA neurons with channelodopsin using the CALCA Cree mice generated by Richard Palmiter's lab. So here you can see an in vivo recording. We turn on the blue laser, the blue dot here, at a high frequency. And here's a unit that every laser pulse evoked a spike reliably at a very short latency. So we're pretty sure that this is a neuron that expresses channelodopsin, and therefore it should be a CALCA cell. And so here's a longer recording from this unit. So this is the EEG power spectrum. I think that Emery showed this. So when you see this sort of broad band, high activity at low frequencies, chances are the animal is asleep. But if you see a theta band at about 8 hertz, the animal is either in REM sleep, blue, or awake, which you can tell by looking at the EMG. So you can see that the firing rate of this unit is high during non-REM sleep and low during both REM and wake periods. So this is a summary of all the identified calca neurons. Each line is one cell across the 3 brain states. And we also routinely summarize the firing rates in this 2D plot. So this horizontal axis is the firing rate difference between non-REM sleep and wake, normalized by their sum. And the vertical is between REM and wake. So the identified blue calca units, they're primarily on the right-hand side of the vertical axis. And so that indicates that these neurons are more active during non-REM than wake. But if you look at the unidentified units recorded in the same area, but probably other cell types, they're really all over the place. Many of them are on the left. So these are wake active. So based on this, we confirmed that most of the calca neurons are indeed non-REM active. So this is our first criterion. Now, are they non-REM promoting? So next we did optogenetic manipulation. So here is our standard protocol. Again, we record EEG and EMG, and this is the brain state classification. So we turn on the laser for 2 minutes per trial with a pseudo-random sequence every 10 to 20 minutes. So here's a summary of all the trials and all the mice that we tested. So you can see that when we turn on the laser at time 0, so this is the 2-minute laser stimulation, there is an obvious increase in the probability of non-REM sleep. So these experiments were done during the day. So the baseline sleep, non-REM, is about 60%, and that's true for most of the mice that we tested. But laser stimulation can bring it up to above 80%. And at the same time, there's a decrease in both wake and REM sleep. We also did inactivation through this mutated channeled opsin. So this is an inhibitory opsin, and that caused a decrease in sleep. We also did chemogenetic experiments because optogenetics, you tend to do it short and you look at the acute effect, but chemogenetic manipulation in a way is more kind of natural. So here the GQ mediated activation caused an increase in non-REM sleep. So the open circle is control and the solid is with a CNO injection. And then GI mediated inactivation of these calca neurons caused a decrease in sleep. So based on both optogenetic and chemogenetic activation and inactivation, we know that these neurons are non-REM promoting. So this is our second criterion. So it turns out that these neurons labeled by the peptide calca, they also have traditional neurotransmitters. And in this brain region, about 60 to 70% of them are actually glutamatergic, and the other 30 to 40% are cholinergic. So without showing you the data, I'll just tell you that we think that the sleep effect is caused primarily by the glutamatergic subset. The cholinergic calca neurons, they seem to be the preganglionic parasympathetic neurons that if you activate those cells, they actually cause the pupil constriction, which by the way also happens during sleep, except that those neurons don't seem to play a big role in directly promoting sleep. Also, just for the sake of time, I'm going to skip a bunch of data and just sort of tell you that in addition to calca neurons, we found 2 more populations of excitatory glutamatergic neurons that promote non-REM sleep. One of them expresses the peptide marker CCK. The other expresses neurotensin. So calca and CCK neurons are sitting right next to each other. And in fact, with slice recording, we know that they have reciprocal excitation of each other. The neurotensin neurons are located a bit more posterior. Now, all 3 glutamatergic populations innervate the POA, right? So this is the textbook sleep center. Calca and NTS neurons also innervate this region called the GIV in the really posterior part of the medulla. So these are also GABAergic sleep promoting neurons. And David, I see an arrow pointing to something. Does that? Okay, anyway, so So I should also say that at least we know that the NTS neurons also form excitatory connections with GABAergic neurons locally within the ventral pack. And these neurons also promote sleep. So it seems like these sort of excitatory glutamatergic neurons are upstream of a whole bunch of GABAergic neurons. And then these GABAergic neurons promote sleep by inhibiting these wake promoting neurons. And I think that, again, Emery mentioned that the anesthetics that are sort of activating the or simulate the GABAergic system promote the loss of consciousness. And this might be one way of how that could happen. So it seems like the ventral pack is really an upstream excitatory sleep center that sort of promotes sleep by driving these GABAergic populations. And in fact, the other thing that's true is that the ventral region of the pack is the only region we have found so far that contains both excitatory and GABAergic sleep promoting neurons. And in addition to some publications from my lab, some of these conclusions were also confirmed by a paper that came out of Yu Hayashi's lab in Japan. Okay, so now I'm gonna switch to the second strategy, which is to screen for sleep promoting neurons. So we did this with based on a very simple-minded idea. So we're gonna start with the wake promoting neurons, and so we know quite a lot of them. And then we're gonna use retrograde tracing to find inhibitory neurons that inhibit the wake promoting neurons. So, as you know, according to the textbook version, this is how sleep neurons promote sleep, by inhibiting the wake neurons. And then once we find the inhibitory sleep neurons, we're going to trace one more step back and look for their excitatory input. Because as I showed in the last slide, if you're an excitatory neuron that drive other sleep neurons, then you could be promoting sleep yourself. So this screening was done by Chen Ma, a really talented postdoc in my lab. So Chen Yan's idea was to target a whole bunch of known wake promoter neurons. And she was looking for brain regions with relatively broad GABAergic innervation of these wake neurons. So these are the 7 populations of wake neurons that Chen Yan targeted, including histaminergic neurons, cholinergic neurons, noradrenergic neurons, and glutamatergic and GABAergic neurons. And these are the brain regions with relatively broad GABAergic projection to these wake neurons. So that includes the POA, right, the textbook sleep center. But Chen Yan also identified a subset of the GABAergic neurons in the CEA, the central nucleus of the amygdala, that are both sleep active and sleep promoting. And so in fact, Chen Yan was the one who initially discovered this neuropeptide marker, neurotensin, that turns out to be a really pretty general marker that labels multiple non-REM sleep promoting neurons all over the brain, including the pack that I mentioned just a moment ago. But today I'd like to focus on this third candidate region, the substantia nigra pars reticulata. And so this is kind of downstream of the striatum that Anne talked about this morning. So now, as you know from Anne's talk, that the basal ganglia, which the SNR is the output stage, is really important for controlling somatic movement. So, and yet this region sort of shows up in our screening. So what does a motor control neuron, neural region, what is it doing in controlling sleep? So to understand that question, we decided to take a closer look at the motor activity of these cells. And I should say that this is characterized by Dan-Qian Liu, a former postdoc in my lab. So in addition to EEG and EMG, we also added video recording in the mouse cage. And our collaborators in Beijing actually, oops, I think that, oh yeah, their names are here. So they trained the convolutional neural network that Fei-Fei Li talked about for automatic image segmentation. So this is the original image, and this is where the network thinks that the mouse is, and you can see it's pretty good. So once we have this automated tracking, it's pretty easy to extract some parameters about motor activity. So we looked at two parameters. One of them is translation, which is basically the center of mass of the mouse in two different time frames, and we want to see how much it moved. The other we call total movement is basically the total number of pixels that are non-overlap between the two time frames. So in this case, it's a total number of blue pixels. And when we plotted these two parameters, we basically saw three clusters, translation and total movement. So the first dark green cluster corresponds to local motion. So this is sort of the highest kind of motor activity. The light green corresponds to all the non-local motor movement, including eating, grooming, or some postural adjustment. The third gray cluster corresponds to immobility. But when we look at the EEG and EMG recorded simultaneously, they actually have two different clusters. One of them is really sleep, and the other is just quiet wakefulness. So here I'm showing you the both the EEG delta power and the EMG power across the four behavioral states. So one thing I want to point out is that if you look at the three greenish states, all of them are actually wake states. So we tend to think of their difference in terms of motor activity. But in fact, if you look at the EEG delta power, there's also clear difference, right? So remember, the higher the delta, the slow activity, the less conscious the brain is. Now, if you look at the quiet wake and sleep states, right? Both of them are immobile states. So we tend to think of their difference mostly in terms of, state of consciousness. And yet, if you look at the EMG, there's also clear difference. So the point is that brain state and motor state, they're correlated across multiple behavioral states. And when we looked at transitions between these behavioral states, they're not random at all. So if the mouse start out in local motion, they always transition into non-local motor movement. We have never seen a case where the mouse was running at one moment and then bang, just fall asleep the next moment. You might see that in narcoleptic animals, but in normal mice, we never see that. And if they start out in non-local motor movement, they either go back to running or they go into quiet wakefulness. They also don't fall asleep right away. So in other words, if you sort of place these four states in a single chain, then most of the transitions are between neighboring states. So this is the transition matrix. The diagonal means staying in the same state from one moment to the next. Right above diagonal means going down the chain one step at a time, and below means going up. So the only exception that we saw is that sometimes the animal would wake up from sleep and directly go into movement and skipping quiet wakefulness. So that was a behavior. And what about the SNR? So it turns out that there are actually two subpopulations of GABAergic neurons in the SNR. If we inject a Cre-dependent virus into a PV Cre mouse, then we're mostly labeling the PV positive neurons in the lateral SNR. But if we inject the virus into a GAT2 Cre mouse, we mostly label the medial SNR neurons. So we call these the PV versus the GAT2 neurons. So next we recorded from the PV neurons. And so again, using optical recording from the tag neurons. So here's a summary. Now, most of the PV neurons are actually most active when the animal has a lot of motor activity, and they're least active during sleep. So these neurons are not sleep active. But when we recorded from the GAT2 neurons, they tend to be most active during sleep and less active when the animal is running. So this is just a superposition of the population averages of the two populations. So when we optogenetically activated the GAT2 neurons, we saw a huge decrease in different types of movement and a significant increase in sleep. But activating the PV neurons has no effect on sleep. The main thing that we saw was a decrease of movement and transition into quiet wakefulness. And when we inactivated the GAT2 neurons, we saw a huge increase in movement and decrease in sleep. And PV neuron inactivation, again, has much less effect. So quick summary, the PV neurons are movement activated, right? So they're most active during movement, and yet their activation mainly terminates movement. So we think that this is because the PV neurons might be important for action selection that Anne talked about this morning. And the idea is that every time we decide to execute a particular motor program, we also need to suppress all the other competing programs, and the PV neurons might be important for that. And this is why whenever the animal is moving, there are always some PV neurons that are active to suppress the other activity. And yet, if you activate all the PV neurons, which is what we did, then you terminate all movement. The GAT2 neurons, in contrast, are movement suppressed, and their activation suppresses movement.
You have 5 minutes to go.
Oh, okay. So I'll try to go quickly. So one thing is that even though activating GAT2 neurons promotes sleep and suppress movement, it doesn't cause artificial transition. In contrast, so instead, what it does is to bias the probability of the natural transitions that we see. So in this case, it's increasing the downward transitions and decreasing the upward transitions. And I'll skip this comment, and inactivation causes the opposite trend. So we also looked at the projections of these neurons, and it turns out that the PV neurons mostly project to the motor control areas, but the GAT2 neurons, in addition to the motor areas, also project to these neuromodulatory wake promoting centers. And using a rabies virus trick, we show that it's in fact the same populations of neurons that send axon collaterals to these motor and brain state control regions. So we also did one more tracing to look for the upstream neurons of these SNR neurons, and we found the STN. And it turns out that these STN neurons also express neurotensin, and their activation also causes sleep. Okay, so I just told you that the basal ganglia circuit promote sleep, and they control somatic motor activity. But during sleep, there's also a slowdown of the autonomic motor activity. So I want to spend the next couple minutes to tell you one more circuit that controls the cardiovascular system, and that's the baroreflex circuit. So baroreflex is a quick negative feedback. The increase in blood pressure sensed in the periphery is transmitted to the nucleus of the solitary tract, and that triggers changes in the heart and the blood vessels to decrease the blood pressure. The circuit is well understood. One pathway is from the NST to the ambiguous nucleus. So this is a cholinergic center that are parasympathetic, preganglionic, and so they slow down the heart. The other pathway excites this region called the CVOM, which contains GABAergic neurons that inhibit the sympathetic adrenergic neurons in the RVLM. So this activates the parasympathetic system, and this pathway inhibits the sympathetic system. So Yuan Yuan first tagged the NST neurons that are activated by blood pressure increase. And so again, using the trap method, and without going into the details, we were able to find them. And then we recorded from these trapped neurons, again, using ultra recording. So here's one unit, and you can see that the firing rate pretty much follows the blood pressure fluctuation. And if you zoom in temporally, a lot of these PE trapped neurons actually are time locked to individual heartbeats. And so that's because they're sensitive to the blood pressure oscillation within each cardiac cycle. So next, we activated these trapped neurons. And so you can see that in addition to decreasing the blood pressure and heart rate, that also increased non-REM sleep. And chemogenetic activation has the same effect. So then we looked at the downstream target, the CVLM. So their activation, again, increased sleep. And this is partly mediated by inhibition of the RVLM. So these are the sympathetic neurons, because their inhibition also increased sleep. In contrast, inactivation of CVLM decreased sleep, and activation of RVLM also decreased sleep. And also we tried to activate the ambiguous nucleus. These are the cholinergic preganglionic neurons, and their activation also increased sleep. So in conclusion, at the beginning, I told you that falling asleep is associated with reduction of both the somatic and autonomic motor activity, in addition to change brain state. So it makes sense for the sleep neurons to inhibit the arousal system and these two types of motor systems. Now I showed you the STN-SNR pathway in the basal ganglia that is part of the somatic motor control that promotes sleep. And I also showed you that the nucleus of the solitary tract, I just showed you, and the beginning I told you the PAG. I also mentioned the amygdala and hypothalamus, right? So all of these structures contain sleep promoting neurons, and all of them are key nodes of the central autonomic network. So instead of being separate as implied by this model, it seems like the sleep control circuit deeply infiltrates both the somatic and autonomic motor system. So I moved the yellow circle into these two motor boxes. And on the other hand, there might be a separation between the specific action neurons from the general state neurons. So in the SNR, the PV neurons might be more for specific action. The GAT2 neurons are more for the general state control. And the general state neurons seem to send axon collaterals to the arousal system and the motor system. So we think that this kind of view of how sleep is controlled might also have some implications for the question of why we sleep. So we don't know what it is, but it seems like the sleep circuit is so deliberately associated with motor control. And that suggests that one function of sleep is to sort of suppress motor activity, probably to promote functions that are incompatible with movement. And so that's something that we're looking into. And finally, these are the people in my lab who actually did work. I think I mentioned their names, and I'm running out of time, so I'll just finish here.